Welcome everyone to this week's coffee morning. It's great to see so many people here interested in orchids and mycorrhizae. So very much looking forward to that. So just a quick reminder of one event that's happening next week that Will asked me to flag, and that's at 12 o'clock next Wednesday. There's a tour going to UCD Greenacre. So it's meeting here and then walking over, I presume, to UCD Greenacre for a look around. So if any of you haven't been, it'll be a really nice opportunity to get over and see all the things that are going on over there. And there's lots of other nice things happening, which are all in the email that's been sent out. But it's my job today to introduce our speaker, who is Aisling Reilly. Aisling is an assistant professor in the School of Agriculture and Food Science and Horticulture. She's originally a plant pathologist, but is now exploring lots of different things. And we're really interested to hear what Aisling has to tell us today. So, Aisling, over to you.
Thank you so much, Dara. And thanks to everyone for coming along. It's really great to see all the interest in orchids. It's very weird for me to be kind of on this side of the coffee mornings. I actually did my PhD and my postdoc here. A little dark time in my life. But it's usually I would be hiding down the back eating all the cake and stuff. So it's kind of really nice to see how life throws you so many curveballs that now I can't eat cake and now I have to actually do the presentation. So thank you all for coming. My talk today is on Mycorrhizae and Shine, orchid secret helpers. And this in itself, and again, it's going to be very kind of more fun facts, preliminary kind of research that we're sort of doing between ourselves and in the School of Agriculture, as well as our colleagues down in Set 2 in Waterford as well. So just to introduce our main event, so the orchids themselves. Orchids themselves are part of one of the largest kind of flowering groups in the kind of the angiosperms or angiosperm family. So they're only second to daisies. There's about 28,000 species of orchids globally. They reach all different types of habitats, of course, except Antarctica, where they can't grow. So very diverse group. A lot of people are very kind of interested in them. They're very unique in regards to like their flowering shape and their life cycle in themselves. In Ireland, we have about 31 different species, with one of them being solely present or native to Ireland, which is the Western Marsh Orchid, which is this guy here on the right. So again, very specific to kind of Ireland and Ireland's landscape as well. But despite being so diverse and being like a very large kind of family of plants, they're actually not incredibly rare, but they're actually very, very specific to the areas that they're sort of growing in as well. And they're very, very sensitive to things like ecological changes. So everyone always seems to be very invested in orchids when they actually have them, because if they appear on land or in certain areas, it means that we're doing something right, that they're there, they're happy, they're growing. And when we lose them from the areas, that's when we start doing things wrong. And so part of our research here in UCD is actually trying to figure out, well, first of all, why are they so sensitive? Why are they so rare? And by understanding kind of different parts of their life cycle, we'll be able to actually help in different kind of conservation programs with them as well. So of the 31 species that are in Ireland, about 6 of them are actually on the threatened list. And most of this is actually due to things like habitat destruction, climate change, tourism, illegal trade, so like the CITES initiatives to try and prevent that as well. But another key thing that actually makes them so difficult to actually propagate and to grow is actually entirely based on their seeds. And orchid seeds themselves are actually referred to as what we call dust seeds. So I've got a little petri dish full of them here. The seeds are not the chaff that's at the bottom. It's actually those individual dusty bits. I'll pass this around so people can see it. All that little individual dust, brown dust, those are the seeds. And they're actually, I'll pass that to Connor to pass around. And they're so tiny, they're actually one of the smallest seeds in kind of flowering plants, and that's kind of on purpose. So the orchid produces millions and millions of these really small dusty seeds that can actually spread far and wide as far as they actually can. But because they're so small, the seed itself actually only ever contains things like an embryo. So this part of the seed is actually contained in this sort of seed coating. It only contains the embryo, which actually then goes on to form the root system and then subsequently the plant. But what a key thing that most seeds need is a food source. So they can't actually germinate if they don't have the energy to actually put into sort of root production as well. And I've included this cartoon here at the side. So this is just a general kind of wheat seed. The germ is actually the embryo. That's what actually germinates to form the root. And the endosperm is actually the food source. So that gives the seed enough energy to actually germinate and form itself. But if orchid seeds don't have that, they don't have the energy to actually germinate themselves. So how do they germinate? They typically do it, and they've actually evolved this system. I didn't think the telly would be this big that you could see how crap the photos are. But this is just some of those seeds that I'm actually passing around in their seed coat. And how we're actually trying to trick them into germinating is we're trying to get them to form relationships with mycorrhizae fungi, or sometimes bacteria as well, depending on the species that we're actually working with. And the same thing happens in the wild, that when all these orchid seeds are spread, hopefully they'll land on a soil profile that actually has the microbe that it can actually form that relationship with. And the fungus basically comes along, it enters in through the pelletone through the entrance of the seed here. It kind of mingles with the seed a bit and actually gives it or helps it provide it the energy that it actually needs to subsequently germinate as well. And so without these interactions, then we wouldn't actually get the orchid plants that we see as well. So if the seeds happen to land somewhere where they can't form that relationship, they won't germinate. What we have the issue is that nobody actually knows which microbes that orchid seeds actually form the relationships with. We actually know very, very little about the orchids that are actually present in Ireland currently. So part of our research here is actually to try and figure out, well, do we have microbes that could form that relationship? Can we actually start germinating and actually help conservation efforts in regards to the orchids that we have? And a very fun fact about this, so the first time this has actually been discovered was actually it's accredited actually to Glasnevin Botanical Gardens up in North Dublin. And we initially found this interaction with this guy, David Moore. Has anyone ever heard of David Moore? Some of the Irish pathologists should probably know about David Moore. He was the director of the Botanic Gardens back in the 1800s. And we know from Ireland in the 1800s, we were going through a very rough period known as the Irish famine. And the Botanic Gardens were actually responsible for doing the majority of the research on this failure of the crop and to actually figure out what happened. And it was David Moore that actually determined that the famine was actually caused by an individual organism. At the time, he correctly identified it as a fungus. And he actually started working his way towards coming up with a treatment in order to try and prevent more infection from actually happening as well. So when he's not trying to save the Irish population, he was also big into kind of orchid research. And he's the first one that published about getting wild orchid seeds to germinate. And how he managed this is that he collected orchids from the wild within their own soil profile, and he sprinkled their seeds very close by to the actual adult plant in itself. And then he noted that they were germinating. He tested this against different types of soils, couldn't get it to work, and actually eventually realized that it's a plant and microbe interaction between them, that they need to have the same species as the parent plant or what the parent plant was interacting with in order for them to actually germinate as well. So again, Ireland is kind of one of more forefronts in regards to orchid research. Back in the 1800s, we kind of fell off a little bit, which is why we're actually trying to get it back kind of running and in the race back again as well. And as I said, again, this is all very, very preliminary. We are doing a few different things. I do actually have a collection of orchid endophytes that were collected by a colleague who's now working down in Set2 down in Waterford. But we're testing them to see if we can actually get that tray of orchid seeds actually germinating and see if we can actually try and correspond some of these microbes and let's see if we can actually get some plants growing. So initially what we've actually got is oat media agar. And we're trying to basically co-inoculate the seeds, and they start off this is about a week on the plate. Again, not a great photo of it. But we start off about 7 days after inoculation. You can see, we can actually see the fungus kind of coming in and entwining itself around the seed and actually starting to entry. We can start to make out when the seed embryo actually starts to enlarge, which is a very good sign. But then eventually we get to a stage where we can no longer see the seeds as well. So these have been growing for the past month and a half. And we get to the point where, again, we can't actually make out whether or not the seeds are actually surviving in there or not. We will eventually have to sort of filter out the seeds. Hopefully we'll be able to see things like roots and a protocorm actually developing. But But also we're trying to figure out how these fungi are actually contributing to orchid seed germination. And there's a lot of theories in regards to, okay, well, it provides the nutrients itself. So it's things for phosphorus solubilization, nitrogen fixation. Potentially, we think there's actually a compound in the orchid seeds that's actually responsible for actually breaking down the mycelia and actually getting its carbon sources from the actual fungus itself. And another thing that we've actually found is that one of our orchid endophytes seems to actually help with root production. And this was a little bit of research that was done by our master's student last year, Jane, where she was trying to biotize or co-inoculate mint propagation through tissue culture with the orchid endophytes. And you can see, compared to like some of the other treatments, the orchid endophyte massively produced a load more roots on the mint plant compared to all the other treatments. So that kind of tells us perhaps that one of these orchids is actually very responsible for like hormone production, things like IAA or auxins. Anyone usually use rooting powder to kind of collect shrubs or collect new plants? Sometimes all of that rooting powder is typically actually an auxin. So it's something very, very similar to this. It's a hormone that sort of tricks the plant into starting to produce its own roots and subsequently its own flowers or its own plants then afterwards as well. So it could potentially be that perhaps a mixture of all of these endophytes are actually required. Maybe when I open these up, and I can pass these around if you want to come up and have a look at them. Maybe eventually these won't germinate. Perhaps I need to have a co-inoculation thing, but we won't really know until another bit of time. The reason this is so slow is usually we don't notice protocorm development until about 6 to 9 months after they've been inoculated. So it is a bit of an investment, but we really feel that it's actually kind of worth it ourselves to try and figure this out. And then we've also got a small project with myself and Dara and Nolene Smith, also in the horticulture department. There is actually a population of orchids based outside, well, at the back of the agriculture building. So if you're going, probably shouldn't tell you where they are, but if you're going directly towards Rosemount or the creche in that direction, on the right-hand side, as you come to the road, there's like a car park and a little grassy bank. And this area here where the estates have actually started practicing like a no-mow May, or no-mow, they don't mow at all actually, do they?
Once a year.
Once a year in September, I think. And when they've actually allowed this to kind of grow wild and become more of a kind of like a meadow grassy area, we've actually noticed that there is actually pyramidal orchids that are actually kind of starting to grow there. So part of Nolene's research is to kind of get the plant growth rates, see where the population is actually traveling to and spreading to. Dara's looking at kind of the pollinators that are come visiting these plants. And I'm trying to figure out what microbes are actually in this bank that are actually encouraging the plants to grow and actually allowing them to germinate as well. So we do have a little bit of microbiome data that we should be analyzing as well, which should give us kind of like a fuller picture. And then everything kind of coming together should tell us how this orchid population is actually maintaining itself then afterwards as well. So I think that's me. Thank you very much for listening, and I can happily take any questions then as well.
Super, thank you so much, Aisling. And I think it's always so lovely when you're working with a study organism that's also so beautiful. It helps everything, doesn't it? Okay, so does anyone have any questions they'd like to ask Aisling? Yeah, sure.
So thank you for the presentation. It was really good and good to see you again as well. So I heard that the fungus is helping the germination of the orchid seed. Can these fungus help? Is there any other cases this fungus is helping other plants as well?
That is a very good question, but there is this whole idea about like endophyte species from different plants. So one study that I was initially working with Professor Fiona Doohan with this during my postdoc, where we had collected, or her previous team had collected loads of bacteria and fungi from wild relatives of wheat, and then we would culture them and actually reapply them to the commercial wheat as well. And we actually found that they had a number of different benefits. So some of them were quite good for nutrient use efficiency. So they would actually help with fixing nutrients. The main crux of our research there was kind of more pathology. So whether or not they could be used as biological control agents to try and prevent things like Septoria and Fusarium actually infecting the plant as well. So it's a very well studied kind of topic. In regards to these particular examples, so the one I've shown in this one, this particular species is actually an epicoccum, which is, again, actually very well documented and actually having a number of benefits. So it's very popularly used for nutrient efficiency and IAA production, which is the roots. So it's actually not surprising that we're actually seeing these kind of responses from these particularly these orchid species one, but a lot of the time some of them are very well documented for multiple benefits. Yeah. Go, thanks.
Anything else for Aisling? Tamara.
Hi, it was a very interesting presentation. Yes, I was wondering actually, one thing I heard anecdotally is with orchids is that they kind of appear one year and then they disappear, and they can disappear for a number of years and then suddenly they reappear again. Can you say more about that? Or, do you have any idea why that happens?
I wouldn't, I don't know the exact reason. I would hazard the guess that it's probably that well, they're annual, so they die off at the end of the season. And if the seeds are dispersed, then if they're not coming into contact with that microbe that it actually needs to germinate, and then it's not going to germinate. But perhaps something changes in the environment. Perhaps there's a land use change, or I don't, through rainfall or people walking through it, perhaps the microbe is then reintroduced to the area. Or perhaps it reemerges and then it can actually form the relationship then and germinate again. But it could be entirely based on the environment itself, the soil. More than likely it is the microbes, I think, that are actually causing it as well. So if it's not coming into contact, it doesn't germinate, but eventually it probably does. And that's when we get this sort of reemergence kind of occurring then afterwards. Yeah.
Anything from online as well? John.
Just link to that.
Just link to that last point then. So can the seeds persist in the soil for a time, or do they have to be kind of fairly fresh to be able to germinate?
No, I think they can reside well for an extended period of time. Yeah, they're typically dormant until they're activated by the microbe in itself. Now they do, when they land, it's theorized that they produce like sugars or compounds to try and entice the right microbes to come near them. But that also entices other things like pathogens that would actually dissolve them and eat them. So as long as they're kind of like happy enough living and they're not actually affected by anything else kind of coming into it, they should reside relatively easily in the soil, I think. Yeah.
So kind of contrasted to wheat research, what I was doing, what I do and you were doing before, you can't really do destructive sampling with these kind of more rare,
No.
delicate organisms. So I was wondering, how do you get around that in the wild when you're looking at what's going on around?
Thank you for that. So it is illegal to sample microbes or orchids unless you have a license that's from the MPWS. And usually in regards to research, you have to make the promise that anything you take out of the field, you have to put back. Part of the application, the funding application that we've submitted this for, one of the outputs we're hoping is to make like a seed coating or like a seed bomb thing, that if we do get the licensing to collect the orchids and the orchid seeds, we would do that post flowering. So the seeds have set, hopefully they've been spread to where they need to. We just take what we need from the plant that's already completed. And then hopefully when we take back into the lab, we'll actually be able to make a product that people can then just throw on the ground. It's the seed with the microbes that they need, and they can start generating more of the population as well. For these ones, the endophytes that I have, I'm going to blame my colleague in situ, because he's not here to defend himself. And he's the one responsible for actually collecting these particular orchids. So like that, we went over to the site behind Ag here, and we only sampled like 1 or 2 plants at the time after they had flowered, and we just basically collected the tuber part of the orchid and then extracted the endophytes from that directly, which we weren't supposed to. But again, he's not here to defend himself, so I'm going to blame him for now. So yeah.
Right. I'm going to ask you a quick question, Aisling, if that's okay. So orchids are famously widely cultivated in horticulture. You can buy them in garden centres and everything else. What do we know from that side of things in terms of propagation? And is there any kind of work done with endophytes in that side of things, or maybe not at all? Maybe that's not used.
Yeah, very little actually. So the ones that we get in Tesco's or wherever for gifts for people, they're epiphytic orchids. So all of the orchids in the wild in Ireland are terrestrial. They need to have the soil beneath them to get their nutrients. The ones in the shops are epiphytic. They've got aerial roots. They get all of their nutrients from water and from the air. How they're typically cultured is through micropropagation and tissue culture. So this is basically where we take very, very tiny, you can actually take very tiny amounts of the plant, surface sterilize them and stick them in a very nutrient-rich completely controlled petri dish or agar jar, and you can basically get them to grow from like the tiniest amounts of the plant as well. Because as I said, this sort of propagation way is so long, like it can take 6 months. Commercially, it doesn't make sense to actually do it that way. So most of the time you can actually, if you have a plant, you can actually chop it up into little pieces and start generating more and more plants from that there. So anything we see in the shops, it's tissue culture. And in the wild, we're mostly dependent on the fungal interactions as well.
Super. Okay, well, I think in the interest of time, you might have to call a halt there, but I'm sure Aisling will be very happy to answer any more questions anyone has. Thank you all very much for coming along today. Maybe see you at 12 o'clock next Wednesday for the Greenacre tour, and maybe you'll join me in thanking Aisling again. Thank you.